This is TWIS, This Week in Science, episode number 835, recorded on Wednesday, July 28th, 2021. Who wants to CRISPR mosquitoes? Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your head with mosquitoes, Neanderthals, and pterosaurs. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. For some, life is a brilliant chance to explore the unknown, to take on the challenges of collecting data, forging an understanding of how things work, and in doing so, seeing the world without the veil of human bias. But for most people, information is rooted in what they believe, not what has been rigorously confirmed. The Yale University poll conducted late last year shows that the majority of Americans believe global warming is happening, 72%. We'll ignore the other 28% for now. Actually, let's just ignore them completely. It's not the only thing they're going to be ignorant about, and it's only a matter of dreadful moments before they segue into talking about the other ignorant beliefs they are metastasized with. More interestingly, is that while not 72% believe global warming is real, only 55% believe that most scientists think global warming is happening. Which is an astonishing reveal. It means that 17% or more of Americans who believe global warming is happening think it's the scientists that still need convincing. If there was only some way to reach them. Further, it found that only 57% think global warming is caused by human activity, which means then 15% of Americans could think that global warming is happening, that scientists haven't figured it out yet, and that it's just a natural phenomenon. Information, assumptions, and conclusions based solely on what they believe. And it's an important note here that while climate change is often framed as a political issue, it isn't one. It just seems like it, because 100% of elected officials in the U.S. who deny climate are Republicans. And while they are only a slim majority in that party, they are the majority in power and hold tremendous influence on what many people believe. While all of these numbers are the way they are because maybe it's the 25% of Americans who reported only hearing something about global warming once a week, what all this suggests is that to get real climate action, all we need to do is convince the media to cover it so that more scientists will believe in it. Because once scientists are on board with something, the next thing that happens is This Week in Science. Coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about science. Like every time I say that, I'm like, I'm going to say something else. And no, it's science because that's what we talk about. This past week in science, what happened? A lot of things, and we're going to talk about them. I have got the mosquitoes. I brought the mosquitoes to the show this week. I apologize, but I think some people might appreciate this story. Other people might not. We may talk about it a little. Additionally, I've got some more folding and some human genome news, as well as a story toward the end related to baby mouse dreams, because those are interesting. What do you have, Justin?
I've got Neanderthal blood. Bees on caffeine. Bees on caffeine. Oral biome history. And some really, really bad news.
Oh, are we going to just do that first?
No, I have a whole thing where I'm going to end my portion of the show, at least, with the worst possible news. And then we'll go off to mouse baby dreams.
Okay, we will go off to I like that. We will go off to Mouse Baby Dreams at the end. All right, Blair, what is in the animal corner?
Well, I have a story about dogs that know you're lying. I also have baboons and FitBits and baby pterosaurs.
Oh, we've got babies on the show today.
Yeah.
Yes, we brought the babies. If all of you out there are listening with your babies and you want to make sure that they hear the show every week, know that you can subscribe to This Week in Science. We are a radio show, but we are also a podcast. You can find us all places podcasts are found, and you can find us on YouTube, Facebook, and Twitch. We are also TWIS Science on Instagram and Twitter. Our website is twis.org. All right, now let's dig into the science. I'm just gonna go straight to that pointy little proboscis, and we're gonna talk about mosquitoes. We'll just start right there. Mm-hmm. With the reality that scientists are working very hard to try to get rid of them. Mm-hmm. They are. And we have talked before on the show about the idea, the new technology, the evolving technology, which has made its way to the forefront in animal research. It's called gene drive. How can we put a gene into a species that takes over and doesn't let other variants of that gene exist anymore? So that in the case of mosquitoes, you put a certain gene into the mosquitoes and they can no longer bite their prey because it changes the shape of their face, basically changes the shape of their face. So they can't bite their prey to transmit the parasite that causes malaria and so many other viruses and diseases. Additionally, they can CRISPR in a gene to make it so that females can't lay eggs anymore. By doing this, if the gene takes over, suddenly you have an entire population of mosquitoes who can no longer bite their prey or lay eggs. And what is the outcome of this kind of a population? Death! They die, not us. Mm-hmm. The mosquitoes die. Mm-hmm.
Well,
so the goal is to reduce mosquito populations and reduce disease in humans. Yeah. But what was your point, Justin? What do you want to say?
Well, I was, from if there was one thing that I would just remove from the planet without any sort of regret, it's always been mosquito.
Yeah.
However, I then learned that there are major pollinators. Mm-hmm. And now I think it's a really, 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 really bad idea.
Mm-hmm. They are major pollinators and they are a major food source for other species. So if we get rid of them, what happens then? So with the gene drive, you hope that something like this will stay within a particular species and that it won't jump between species.
That was what I was going to ask about.
Yeah. So far, we don't know the answer to this question, but the study that is out this week in Nature has achieved gene drive in populations of mosquitoes in a lab to kind of virtually real scenario. So we're not talking about mosquitoes in little tiny aquariums. They have built climactic chambers that are supposed to have more realistic environmental aspects to them. And this is part of the process of testing gene drive. The question is, you can make these things happen in the lab, and you might get to 99% suppression of other gene variants and 99% effectiveness of the gene that you've stuck in there, that's driving its way through the population, making its way through 99% of the population in the laboratory. But what happens out in the wild? What about the individuals that get eaten? What about the individuals that just die? What about the individuals that just can't handle the cold? How do their genes, do things change when you put them out in the wild? And so they stuck them in some very large feeding cages and showed that, lo and behold, when they put the gene drive in these populations, it still worked. So laboratory to realistic-ish situation, it works. Gene drive still continues.
So, the whole idea of gene drive jumping from mosquito to anything else seems to me implausible. Like, not just like, it might not happen, but like, I don't even know how you could create that to make it do that. Like that, you know, I mean, like to even engineer something that you would want to do it would seem impossible. So I don't think that's a fear. But you're like, why all of, like you're saying, all the things that feed off of mosquitoes.
Right.
Bats,
they didn't have any of that in there. There were no bats.
No.
No bats in these cages. Yeah.
But then it's sort of, if we can do this, why can't we give them like the butterfly proboscis? Something where they can change their diet and eat something else.
Right.
You know, transmorgify them into a creature of our own design.
Just selective breed them, basically. Just make them a much less dangerous version.
Yeah. I mean, I think if we could, these are the bioethical conversations that need to take place. Which, yes, this seems to be working. But what happens when it does actually get into the field? What exactly does that mean? Knowing that that this bridge situation with the large field-like cages for this paradigm of studying gene drive, not in the field, but close to it. And then we're going to learn more about how the gene drive works in this situation. I mean, that's good. It's the ethological way to study this, but we still have to have these questions of, is this the kind of gene that we want to change population structures? Is there some other gene that we should be working on? Should we just be affecting feeding behavior and not ability to produce offspring? Should we, are there better technologies?
So, and the real ethical one, way to go would be to alter our own genes so that we were not appeasing a meal. That would be the only ethical way to do it. Is as a species we would decide to change ourselves, deciding to make this whole change for environment and altering another species. It sounds, again, until I found out that there were pollinators, I didn't care. Some bats went hungry. Pick another bug. There's other bugs out there. You didn't get them all. but now I'm worried, like, how we don't necessarily, or we definitely don't have an idea of how important they are to our biome environment overall.
Yeah, I think the big question for me is we've seen a lot of gene flow between species of lots of different kinds of species, but mostly plants. But it does happen in insect species as well. And if species, we think of them in these little boxes, but really there's this continuum of one species into another, and with subspecies in between. So if you have subspecies interbreeding, that could lead to this kind of a gene driving through various populations and getting into different species, then that's the big question. Will releasing something like this into the wild have an effect we can't take back later? And we really need to answer that question. But
and I think it's
trying
still
to do the experiments. But go ahead.
Yeah, and I think it's still a really important thing to explore because malaria is extremely dangerous. That's not the only thing that mosquitoes spread too. And we are already messing with ecosystems due to climate change. So as much as I want to keep things how they were, mosquitoes are now going where they weren't historically because of climate change. So even though I want to keep pollinators, and that's an extremely important thing to keep in mind, you also have to recognize that, like, this is kind of a runaway cart with all sorts of things. And so maybe the spread of disease through mosquitoes in places where they're not supposed to be because of climate change could be worse than potentially losing the pollinators themselves.
Yep.
So there's it's complicated. I'm just saying it's I know I'm usually the one saying, like, keep everything the same, but this is one of those situations where I recognize it is extremely complicated and this research needs to be done.
Yeah. Yeah, I don't think we should stop the research. I definitely think it should continue, but I think it needs to be done with continued conversations with communities, and we need to have these conversations to see, what do we think about this? What do we want to do? What's more important?
Very interestingly, I'm getting word from the science line here there's a Danish malaria vaccination on its way where the antibody is transferred to the mosquito when they drink the blood, and they have found out that it can kill the parasite in the mosquito. So now you have...
That's impressive.
Now you have a vaccine that prevents you from getting sick, but also is curative to the mosquito. That sounds like a better strat. You might want to focus.
I'd like you to focus on Neanderthals right now.
Okay, so...
And their blood.
Yeah, Neanderthal blood. So, there's not a whole lot of samples out there, right? Before genetics was a thing, one of the ways in which anthropologists could try to determine the origin and migration of human populations was through blood typing. When genetic data became available on humans, blood typing became largely relegated to making sure that you got a successful transfusion. Now the two techniques are being combined in a way to sort of look back into the history of Neanderthal and Denisovan evolution, health, and some of their vulnerabilities based on their blood work. Now, how do you get blood samples from a 40,000-year-plus old Neanderthal?
Ask nicely. Give him a lollipop.
I don't know. You have to, I don't know.
I have to tell
thaw
you?
it, put it in the microwave? What?
Aha, so they haven't, so these fossils, and there has been like, yeah, we could get some recognition that there was heme and like some very old bones and stuff, but no, they didn't get a blood sample. What they did was they got enough of the genome decoded that they can look at the genes that code for making the blood. And within that, they can see the alleles, they can see the types, they can see the blood through just the genome. This is Sylvana Condemi of the Centre National de la Recherche Scientifique and her colleagues in France investigated the previously sequenced genomes of one Denisovan and 3 Neanderthal individuals ranging from 100,000 to 40,000 years old and managed to determine blood types and analyze the implications of what was there. So there are some, I guess there's more than I thought. There's like, it says here, 43 different systems for assigning these blood types. And they looked at the sort of most common types first. And they found that Neanderthals and Denisovans are polymorphic for ABO, which is like current modern humans have A and B and O blood. That's so different. And actually they shared, these blood groups also shared alleles that are recurrent in modern sub-Saharan populations, therefore making it consistent that the idea that Neanderthals and Denisovans have African origin. Something that has not been hugely argued against, but there have been some indications or some talk that maybe Denisovans could possibly have a non-African origin, or at least not as recent. But it looks like, yep, they're consistent with sub-Saharan populations in terms of blood typing. In addition, a distinct genetic link between Neanderthal blood types and the blood types of an Aboriginal Australian and indigenous Papuan suggests the possibility of mating between Neanderthals and modern humans before the modern humans migrated to the Southeast Asia area. Neanderthal individuals also had blood type alleles associated with increased vulnerability to diseases affecting fetus and newborn, as well as reduced variability of many alleles compared to modern humans. The pattern, they say, is in line with existing evidence that we've also found from other things that links low genetic diversity and low reproductive success with the potential demise of the Neanderthal population. They also found genes in both Neanderthal and the Denisovan associated with strong resistance to norovirus. Can cause upset stomach. And also just goes to show viruses have been everywhere and we've been dealing with them forever. Not new. Yeah. So it confirms African origin, some weaknesses in their fertility, and a little bit of virus fortitude amongst our ancestral cousins. So this is published in the Public Library of Science online. So if you want, you can go check it out. That's free.
That's awesome.
Your local library of science.
All right, Blair,
Yes.
What was your next story?
It's just a little story about dogs. So dogs, they are pretty intuitive. You think, they might know a little bit about what's going on in your brain based on your body language and what they know about you. And so this is a story that I brought for the quick stories because it's an excellent headline. But I think I really wanted to just look at the experiment itself and talk about it for a second. So the headline is that dogs can tell when people are lying to them. This is from the University of Vienna, and they claim that they can indeed tell if a human is being an unreliable deliverer of information, if they are lying, if they are being knowledgeably obtuse, you could say. So they did a study with hundreds of dogs, and they were taught to follow the advice of an unknown human in choosing which of two bowls contained a hidden treat. This is very similar to stories we've talked about before on the show, where they just kind of pointed a bowl, and that is the bowl with the treat in it. By following the advice, they get the treat that's in the bowl. Then they allowed the dogs to watch as another unknown human moved the treat from one bowl to another, while the second unknown human watched. So then they were able to see that this human knew that it wasn't in the bowl that they were showing them. They also did one for control where the human didn't see where the treat got moved to, and then they came in and pointed at a bowl. And so then they conducted the same experiment. Which bowl will the dog go for? The dogs ignored the human advice if they weren't in the room. So they appeared to not care at all about what bowl they were pointing at because they weren't there. How could they know? So that's interesting. I think that's actually the interesting piece of this experiment, is that they could identify what knowledge that human had. But what they claim is kind of the crux of this study is that when the human knew where the food was and pointed at the wrong bowl, half of dogs ignored them. So that to them means that the dogs knew the humans were lying. I would say that means the dog knows where the food was.
Well, either the dog knew where the food was, but I mean, half, 50%. That's like chance.
Yeah, exactly. That's my other problem.
That's random. That's like, yeah, could be either way.
The other thing that's interesting about this is that they then compared it to similar research with humans under the age of 5, macaques, type of primate, and chimpanzees. And in those experiments, the children and the primates were more likely than dogs to follow advice of an quote-unquote obvious liar over what they knew to be true. So what they're saying is this suggests that the dogs were less trusting of unknown humans with advice. I also think that's kind of a leap. I would also like to know whether they used treats that smelled at all. I would like to know if they did this again with a trusted source, how they would act. I think that this is an interesting first step to recognize that they behave differently than other animals in previous experiments. I think that's really what's interesting.
That's interesting.
And I think it's interesting that they knew, like, you don't know anything. Let me figure this out for myself. I think that's really interesting. I don't know if this really points to dogs knowing when humans are lying, but I think it does mean that dogs know what humans know if they're there to see it. So it's almost like this is a reach, but it's almost like they can put themselves in the human shoes and be like, okay, I know what that person saw, so I know what information they might have. It almost implies object permanence, which I think is really interesting, but that's a whole nother thing. Anyway, dogs are smart. They're social. They understand how to communicate with humans, and they might be better at that than wild animals and babies.
Great. Might be. More study needed.
Yes.
For sure.
For sure.
Larger sample size.
Yeah.
Yeah. Let's not use treats. Let's use food. Let's use toys that don't smell.
Yeah.
Yes. You want a dog that is not as smart as your child.
Yeah.
Yeah,
because they don't speak English, so it makes it really hard to reason with them.
It does. Don't worry about reasoning with your dogs. No, that just goes down it's bad paths there. Speaking of reason, artificial intelligence has a lot of it, essentially. following up from last week's story about the Rosetta Fold open source protein folding predicting AI that was released. Basically, at the same time, AlphaFold came out and made its source open to the public as well, so that people could peek behind the hood and see how it worked. Additionally, they published that they were predicting the structure of more than 20,000 human proteins, in addition to almost all the known proteins from 20 model organisms: E. coli, fruit flies, yeast, soy, rice, lot of organisms, mice. They're going to add to this database that's going to be a publicly available, searchable database. It's a combined total of over 300,000 protein structure predictions. The company has released all this online. It's going to be held by the European Bioinformatics Institute, based at the European Molecular Biology Laboratory. And as of last week, researchers were going into this database and searching for their proteins. Biologists looking for the proteins that they study to see if the structure had been adequately predicted. Well, it's not that great. It increases us from our previous amount of 18% of the human genomes, or the human proteomes of the human, 18% of the human proteome, to about 40%. We now have pretty accurate structures, very accurate structures, for about 40% of the human proteome. Now that's a lot, but that leaves a lot of proteins still up for grabs. And while I was snooping around on Twitter last week, I saw a lot of researchers posting images of the proteins that had been, the structures that had been predicted for their proteins, and just shaking their heads and laughing and going, "This is telling me nothing. This is totally wrong." And so a lot of researchers are still going to be waiting for their proteins to work. And the reason behind a lot of this is because there are a lot of proteins that they only get into their final conformation when they interact with other molecules. So because we don't know what the other molecule is necessarily, or we don't have that information involved in the artificial intelligence, the deep learning that would predict all the structure, the end structure, there's no way to know what it would look like, because the structure is going to be determined by some other forces, molecular forces. But that said, opens up our catalog of protein structures massively, adds to this ability for researchers to be able to link together structure and function and understand a lot more about us and the world we live in. And it's just the beginning. It's just the beginning, which is so exciting. But some of the other proteins that are not included in this catalog are also larger. So they're too big, and AlphaFold set a cutoff for the size of the protein, the number of amino acids that would be included in the proteins.
The bigger it is, the more crazy different ways it could fold.
Yeah. So researchers working on the big giant proteins, you're out of luck, but there are fewer of you. So, yeah.
I know.
Whatever.
I picked the wrong protein to research.
It'll happen eventually. You'll get there.
Don't give up, science.
Don't give up. What do you have next, Justin?
Bees on caffeines. Yeah, so there's been past studies that showed that bees like caffeine. They will more frequently visit a caffeinated flower, or one that is treated with some caffeine. And these researchers thought, okay, that's interesting. They seem to like caffeine, but maybe this is just something that's going on within the bee itself. Maybe they're showing up more to that flower because it's also more interesting, or they can smell it better. Maybe it's affecting the bee's memory of where that flower is.
Isn't caffeine addictive? So
no, why would you say that? There's a Starbucks on every other corner now, but that doesn't mean it's addictive. It just means it's really, really good. You have to have it to start
So they're
your
just
day,
getting
otherwise
bees
your day is terrible.
hooked, is what's happening.
Well,
Hooked on caffeine. Well, I mean, caffeine is supposed to enhance learning. It's supposed to be this drug that makes you more attentive. I mean, at least more excited or excitable or caffeine.
well, and that's exactly
Only had
right.
my normal 2 cups today, I promise.
That's exactly the effect that they ended up discovering. So when you give bees caffeine, this is Sarah Arnold, who's a researcher at the Natural Resources Institute, University of Greenwich in the UK. When you give bees caffeine, they don't do anything like fly in loops, but do seem to be more motivated and more efficient. We wanted to see if providing caffeine would help their brains create a positive association between a certain flower odor and a sugar reward. It's really quite challenging environment out there for bumblebees because they don't have extraordinarily sharp vision at long range, according to Arnold. They need to rely on a lot of cues, such as their sense of smell, to find good flowers. So instead of dosing flowers with the caffeine, what they did was they went to the bee box, they went to the hive, and they created a mixture of sugar, caffeine, and this scent of I think it's some kind of a strawberry plant flower. Where's it at? Anyway, some sort of strawberry. And then they watched where the bees went. And they had some trained bees, some untrained bees. They had all these groups, and they went out and tested them. It turned out the 70.4% of the caffeinated bees visited the strawberry flowers first, which was much higher than chance. It was compared to 60% of the bees given just the strawberry odor and sugar, but without the caffeine. And it was almost 44.8% of the bees given only sugar flew to these strawberry flowers. So the suggestion was the caffeine did give a noticeable edge to the bees' ability to recognize strawberry flower from its odor and to remember the desired nectar. Now, this died off pretty quickly because they still had the sugar was still back in the hive. So every time they went back, they still got sugar, and then they eventually we'll just go anywhere. I should just stay here. This is fine. I don't even need to go out now. But they think this could have an implication for training bee boxes for farmers. Farmers, I think there's some crazy number, like 80% of the bees in America come to California for the pollination season. And so you see these bee boxes out there in the fields next to the orchard or the sunflowers or the strawberries or whatever's out there growing. But if the farmers can then train their bees to prefer their crop and maybe give them a little caffeine so they'll be better workers. This is why every office should have coffee maker in it. Every place of work should be providing free coffee. I know
and
you're
allow
thinking,
coffee breaks.
I know you're thinking, hey, I don't want to pay for coffee for all of my employees. But the thing is, they'll be more efficient, enthusiastic, and frankly, just awake. Mm-hmm. I mean, farmers are going to start giving it to bees for free. So,
well, Justin, if you give a bee coffee, they're going to ask for milk. And if you give a bee milk, they're going to ask for sugar. And if you give a bee sugar, they're going to ask
for a cookie.
For a scone. Yeah. And if you give me a scone, they're going to ask for gel. Should I keep going?
No, that's okay.
That's fine. That's fine. Bees can have all those things. What do you got against bees having scones?
Honey. They have honey with scones. There you go. Perfect. Full circle.
I love it. We've known bees are very intelligent for a really long time. So it's interesting to see that a compound that is known to accentuate these engagement, attention, kind of learning behaviors in humans, that it has a similar effect in an insect like a bee. So now, drink your caffeine and be busy as a bee.
Get nice and buzzed.
Mike Shoemaker.
That is the topic. The caffeine, though.
Wins
I was going to say
puns.
Does he?
Bee-rista?
Yeah.
But Michael Pollan has
better than that. What was it?
an audiobook all about caffeine. So this fits in here 100%. I have news for everyone. Big news.
Yes.
The human genome has been sequenced.
Again? Wait, what? Didn't we do this?
That's so
or is it just
oh.
like, did we just do a rough draft the first time?
Yeah, didn't it finish in the, I want to say late 90s we finished it, right?
No, it was early 2000s.
Oh, okay.
Yeah. 2013 was around, 13 was around when they announced the completion, but it had a whole bunch of holes in it. And then around about 2019, some researchers decided that they were not happy with the holes in the DNA. And it wasn't a lot. It was about 8% of the genome. And computational biologist at Johns Hopkins, Michael Schatz, says there's basically an entire human chromosome that had gone missing. So, of course, researchers are like, what's in there? What's going on? And Adam Philippi, a computational biologist at the National Human Genome Research Institute, one of the researchers, along with Karen Maiga, a geneticist at the University of California, Santa Cruz, who founded the Telomere to Telomere Consortium to fix this problem, said, and this is from Carl Zimmer's article in the New York Times, that part of the motivation for such an audacious project was that the missing gaps annoyed him. They were just really bugging me. You take a beautiful landscape puzzle, pull out 100 pieces and look at it. That's very bothersome to a perfectionist. So because there were bothersome holes in the data for the DNA, for the human genome, these researchers pulled together not long ago, 2 years ago, to complete this. And they got around 99 scientists working to sequence what was left and actually sequence everything. They figured out new ways to make sense of the data, to figure out how overlaps were working, and to just to work it through. So it's not just that they filled in the holes by just sequencing what was missing. They did sequence the whole genome, and they put it together, and they ended up finding about 2,000 genes that we didn't know of at all. A lot of them don't look like they work. They look like they have mutations, but there are about 100 or so that might be able to produce proteins, and we don't know what they make.
Important.
Yeah, and so now researchers are looking at this new data, and they're starting to take, instead of matching up their patients' genomes to the old version of the human genome, they're now starting to match up their patients' mutations against this new data to see if there is anything new that comes of it that can help them treat diseases, that can help them understand where certain diseases that are genetically based come from.
We should talk to the guy with the really big protein that he can't get into the AlphaFold, because he's been like, it's a human-made protein. They're like, no, it isn't. It's not anywhere in the genome. He's like, but it's in the humans. They must have gotten it somewhere else. No, I'm telling you, it's really big. It's humans. And now finally, he's like, aha, I've been telling you all for many years.
Yeah.
Nobody listened to me.
Nobody listened. Yeah, they found some weirdness around the centromeres. With the centromeres, that part of the chromosome that kind of moves things around during cell division. It anchors proteins during that division process. And apparently there's a lot of repeated stuff in there, but there's a lot of DNA that they don't understand why it's there, because it doesn't seem to be involved in the centromere's key purpose. So there's a bunch of stuff that's kind of popping up that they didn't know, that now they're getting a little bit more information. And so this is very interesting. So yeah, 2021, we have the entire human genome. Finally. But yeah, pretty good. It's exciting. I mean, maybe someday we'll actually know what to do with it and how to interpret it. That's the next step. This is This Week in Science. Thank you for joining us for another episode of science news and discussion. If you enjoy the show, sharing it with your children, sharing it with your family. Yeah, You can share it with your friends, too. Tell them about TWIS today. All right. I only have a couple of COVID stories this week.
They're not good. They can't be good.
Nope, I just want to follow up on the CDC's reassessment and the fact that they really are saying, yes, if you're vaccinated and you're in an area of high viral spread, then it would be safer for you. They're recommending that you wear a mask in public places, indoors, where there are other people. The Delta variant is about 1,000 times stronger than the previous variant. So there are calculations that have been made that, of course, are being contested because it's not exact. But the CDC had said you could spend about 15 minutes if you're unvaccinated wearing a mask indoors with another person who was wearing a mask indoors. And that was because of the amount of viral particles that are released during breathing and how many particles it seemed to take to cause an infection. They said about 15 minutes and you'll get infected. Well, based on the fact that it's about the Delta is about 1,000 times stronger, that now comes down to about 1 second. You can spend very little time with somebody and still get infected because there are so many. There are 1,000 times more transmissive particles being released with the Delta variant. This is why it is more transmissible. There's just more of it. It's why more people are catching it. It's why more people are transmitting it, because it gets in, just making lots and lots of viral babies inside your nose. We don't like that.
So wear a mask indoors again.
Again, please.
And
because
encourage your friends and family to be vaccinated. Please.
Please. Yes. And apparently there are people who have been suggesting that you don't need to get vaccinated. You can use ivermectin, which is an anti-parasitic drug that is used very well, very often for getting rid of parasites. It has never been used against COVID before, but there was some people said, well, it works here, and there was a little bit of data, and so individuals, because it's a very inexpensive drug, people have been pushing for the use of ivermectin as a treatment for COVID-19. The Cochrane Collective, they review ideas. They go and they review all the literature, and they do these amazing reviews, and are like, people look to them in the field. What did Cochrane say? They did the review, and the Cochrane review has come to the conclusion that based on the data, laboratory tests suggested a weak effect on SARS-CoV-2 virus in a test tube, but did not seem feasible in humans as the doses needed would be large. However, early small trials suggested large effects on mortality, and this has led to some advocacy groups lobbying for its widespread introduction worldwide. And in the published paper, the researchers go on to say, the coordinating editor, Professor Paul Gardner, went on to say that this is a great review from an experienced team. The hype around ivermectin is driven by some studies where the effect size for ivermectin is frankly not credible, and this has driven the conclusions in other reviews. The study with a huge effect has been retracted as fake. Careful appraisal is the cornerstone of Cochrane's work, and with such extreme public demands for a drug to work during the pandemic, it remains vital that we hold on to our scientific principles to guide care. And so the main authors went on to say that current evidence does not support using ivermectin for treating or preventing of COVID-19 unless they are part of well-designed randomized trials. And there are none of those trials here in the United States. Oxford is going to be starting to use ivermectin in part of one of their trials, and I believe there are a couple of other ongoing trials as well. But for right now, don't try and rush things that you do not know, Jon Snow.
My mouth is very small.
I know, I'm watching your mouth, and if the listening audience could see what has happened.
My mouth is very small because I'm very
pursed.
angry because people would rather catch COVID and use experimental medicine than take a vaccine that has been widely tested.
Yes.
It's very frustrating to me.
No, but it's just business. I mean, they don't have a touch. They don't get a touch the vaccine money. But they got all kinds of sideways cures that they got stakes in. That they get a nice touch. So, yeah. Hey, I got a suggestion.
Yeah.
Before we do global pandemic ×2 and need a new vaccine, can we go back to my first suggestion? A year and a half ago? And just take 8 weeks off.
I really could use a vacation. I'm tired of my house.
Everybody.
Yeah.
Take 8 weeks
would be
off.
ideal. Yeah.
Everyone stock up.
Everybody. Otherwise, guess what? It never ends.
Yeah. No, in fact, it gets worse again. Yeah.
Y'all know that this show only happens because of you. This is This Week in Science. Thank you for being here and being with us for this episode. We hope that you're here next episode, and we hope that you were here the last episode. We hope that you stick with us. And if you would like to help support TWIS, head over to twis.org, click on the Patreon link, and then join our Patreon community and choose your level of support. $10 and up, and you will get a thank you by name at the end of the show. And there are some fun little gifts that get sent to you as well. We really can't do this without you. Thank you for your support. All right, we're gonna come back right now to a little part of the show that's called Blair's Animal Corner!
With Blair!
Why do I always turn the volume down?
Bugs are creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
I'm gonna let you pick, Justin. You want pterosaurs or do you want baboons and FitBits?
You had me at pterosaurs.
Okay, great. You got it. So this is a study looking at baby pterosaurs. Yeah.
Who's your little dinosaur?
Yes,
oh, you little dinosaur.
So researchers from the University of Portsmouth and the University of Bristol, along with paleontologist Darren Naish, found something about baby pterosaurs. So we haven't really found, the just science in general, I'll say, has not found a lot of baby pterosaurs, a lot of pterosaur eggs, newly hatched pterosaurs. These things have not been recovered in fossils very much. And so this is something that is more of a recent discovery. The first fossils of pterosaur embryos were discovered in 2004, but they've known about pterosaurs for over 2 centuries. So all that to say, exactly the biology of a baby pterosaur, how they come out of the egg, has been a bit of a mystery to this point. For those of you that don't know, pterosaurs lived during the Triassic, Jurassic, and Cretaceous. That's about 228 million years ago to 66 million years ago. They were around for a long time. And so there was this question of what baby pterosaurs looked like and if they were capable of flight. Because if you think about birds, for example, they spend a pretty long time like wet, gooey, and then fluffy and just completely helpless. It takes them many days to weeks, depending on the species, to be able to fly. So there's this question of, do they come out helpless? It's called altricial. Or do they come out precocial, like a baby cow or a baby giraffe that's like up and running around in an hour, right? So this question was actually answered via fossils. And so they were able to look at humerus bones of these babies, and they found that they were stronger. Yes, the funny bones. They were stronger than that of many adults. So they were strong enough for flight. So then they used computers to model the flying abilities of hatchlings. And they used previously obtained wing measurements from established hatchlings. So these are like juvenile pterosaurs. Then they had the embryos, and then they had the adults. And all of this compared together found that as I mentioned, strength of the humerus bone was more. That was enough that they feel very confident that they could fly, but that they were flied very differently than adults. So these tiny, newborn little hatched pterosaurs were about 25 centimeters long. So that's like under a foot. It's a very small, it could fit in the palm of your hand. Spill out a little bit, but pretty much could fit in the palm of your hand. And they were strong enough, based on this computer modeling, to sustain flapping and takeoff, which is pretty stressful on a body. And they were shaped and powered for kind of flapping flight instead of gliding flight. And so adults do more gliding flight. So they think that parents, the adults, were slower, and the younger... Oh, sorry. I got that backwards. So the hatchlings were smaller, or were slower because they're smaller. So they just get less power, but they were more maneuverable. So the adults, being these big gliders, like a hang glider, it takes forever to be able to turn, right? So they're faster, but they're less maneuverable. So the question is this just the way they come out and the fact that they're smaller and their muscles, I don't know, maybe they're not developed the same, or there could be all sorts of reasons that they fly this way? Or is this actually the result of evolution, and did this enable them to rapidly escape predators because they're so tiny and they're easily preyed upon? So then that leads to other crazy questions we can never really find out, which is, were they independent from their parents? Because if they were, they would need to be more agile because they weren't being protected. Did flight style influence habitat choices? Did they separate from the egg and the parents and go into a different type of habitat or a different niche because they flew differently? Did this change as they grew? So there's all sorts of potential kind of further conclusions you could draw from this research. And so obviously you can never just observe a pterosaur and figure this out, but you have to kind of piece it together with other clues from the fossil record.
Well, the thing that makes me wonder, right, is did the adults continue to be able to fly? Like, did the adults grow to such a size where they're just like, it doesn't even make sense for them to fly, but it's such an advantage for the children to be able to be small and get up into the air away from predators that because the parents are pretty decent size. Like, it's even hard to imagine a pterosaur flying. Like, it's just like, it's almost like that can't be a thing that ever existed big. Mm-hmm. Maybe they didn't fly. Maybe they just came back, that's just a juvenile thing, all the flying.
I think they'd get eaten real quick because they'd be really clumsy on the ground. They'd be lunched real fast.
They have an almost quadrupedal ability to maneuver on the ground. I doubt they're very quick, but they would run into that anytime they were on the ground. I don't know.
And for those of you listening, the chat room is telling me that I should have made very clear I'm talking about pterosaurs.
You just blew up your mic too with the hard P.
Sorry. Pterosaurs. I got my shield up.
It's my fault actually, because I was like, "Oh, he's a cute little dinosaur," and they're not dinosaurs.
I mean, it's anyway. It's with the P in the front, like pterodactyls, but there's a whole family of flying dinosaurs. You should have caught on by now. I was talking about flying dinosaurs, right? Like, I feel like that
flying dinosaurs.
I thought they weren't flying dinosaurs.
They're not. They are and they aren't. It's kind of like saying a spider's not a bug. Okay,
it's like saying Curious George is a monkey. Not a monkey.
No, that's different.
That's the same thing.
No, you're right, actually. You're actually totally right about that. It just hurts my heart so much more. Anyway, so that's the pterosaurs. Moving on. Yeah. Let's talk about baboons and FitBits. So this is a study from Max Planck Institute of Animal Behavior. And they put what is basically FitBits accelerometers on an entire troop of baboons, about 25 baboons. And they wanted to see the detailed movement and behavior and related energetic costs of a troop. This is because baboons live in stable societies of up to 150 individuals. They can be as big as 30 kilograms, which I'll let you guys do that math. I can't do that in my head. I can do some of the other stuff, but not this one.
So 30 kilograms is approximately the same as 30,000 grams.
Very good. Yes.
And it's also, I did a quick Google while you were doing that. Thank you very much for the vamp. That is 66 pounds. So anyway, they can be as big as 66 pounds. They can be little babies. So all that to say, they have this mixed age group. And as you imagine, this is directly related to our last story, that 66-pound baboon and a baby can move at different speeds. So they wanted to see the dynamics of the speed and pace and energetic output of the individuals in this troop. How do you move as a troop of 25 or potentially up to 150 when you have this wide range? Do you all slow down to the slowest? Is there kind of a push to get to a middle ground? Is everyone following the dominant male? What is happening there? And so by being able to strap these accelerometers to all of the baboons, they could figure out the energetic cost and the speed and the relative location of all these guys. They actually studied this baboon troop since 2012. The senior author Meg Crowfoot has been studying them for almost a decade. And so they came up with about 10 million points of data showing where the animals moved, and the accelerometer had 120 million readings, giving ultra-high resolution information on on movement behavior. That's like speed and the number of steps. So while the sample size in terms of individuals was not large, the actual number of data points over almost 10 years is very large. So there is something that you can draw from this and kind of get a nice average, at least for this particular troop of baboons. And they found that the animals do in fact have a preferred speed of movement depending on their body size. So a large male with longer legs will obviously move with longer strides, that nice pendulum motion, require fewer steps to get farther. So they like to go faster. Juveniles have smaller legs. They move slower. Anyone who's tried to walk hand in hand with a small child knows this. So knowing all that and being able to come up with their preferred speed based on energy output at speed and things like that, they then looked at how fast the troop actually went. And it looks like all baboons compromised so that they could match the pace of their nearest neighbors. This sounds to me almost like a school of fish, which seems very cool, kind of population dynamics, and there's all sorts of interesting stuff happening there. But when the group spreads apart, they try even closer. So when they span farther distance as a troop of 25, they kind of jog to catch up with those around them even more. And so this provides evidence of what they consider a democratic process at work. But really, I think it's more just kind of mechanics of a group in movement. And so they specifically wanted to see too what the dominant male was up to, because in one-on-one interactions, dominant males definitely have the ultimate say-so, but in collective movement, they actively slow down to stay with the rest of the group. And it looks like the individuals that sacrifice the most in the group are the smallest ones. So the smallest baboons.
Because they have to keep up, right?
Yes.
They have to work so hard.
Yeah. So they have to keep up to get with the rest of the group. They have to travel longer than they wanted to go, and they put out more energy. They pay a disproportionate cost to stay with the group cohesion. So the researchers say this might be because they have the most to gain from group membership, which totally makes sense. But I also think the youngins can handle more physical stress, and that might also have something to do with it.
And it builds character, even if you're a baboon.
I will say though, yes, you have the young'uns with all the energy. They can keep up. That's great. But they also tire out, and then they whine, and then they end up dragging on you, and you have to carry them. And
but then they also recover the next day, like it never happened. Whereas you are still tired from the long walk at the playground and the thing.
But as a vertically challenged person, slightly, I'm not as tall as some of the other people I know going walking with. I have always had to, my legs, they're shorter. I've always had to move them 2 strides for other people's 1.
Now, did you keep up with those people, Kiki, because you are worried about being predated upon?
It depends. I was going to say no, but then I thought about places where I'd been, and I'm like, well, actually, that depends.
Yeah, walking home late at night is a whole different story. But anyway,
some neighborhoods, yeah.
So the last bit of this study, which I think is interesting, is they used computer simulations to see how the group would look if everyone moved at their preferred pace. What would happen to this kind of dynamic, this flow of the group? And then they compared with individuals adjusting their speed in response to others. And they really showed, to no one's surprise, I'm sure, but the science has to be done, right? That the locomotor compromise is a requirement for cohesion, which just means somebody's got to speed up and somebody's got to slow down if everybody's going to stay together.
Yep. You have to come to some kind of agreed upon middle, and it has to be, At some point, I'm sure, if there is a predator and one of the large males or the sentries sees that male and is like, or sees the predator and is like, everyone get together and go faster, then everybody runs faster together. But it's all about protecting as many of the group as possible.
Yeah. And so last thing I'll mention is about kind of next steps of this study, the implications, which I think is an important thing to mention here. So the differing locomotor ability, the different speed at which animals can move, might put an upper limit on how large a group can be, might force specific individuals to group together, and might have impacts on the structure of animal society. So to this point, there's not been a lot of FitBits on animals in the wild. This is one of the first studies where they've really done this thoroughly. And so being able to study this aspect of cohesion of a group or a troop or a pod or a pride or whatever you want to call it, could have important implications in the future when you're studying the kind of population dynamics and ecology and all these sorts of things. So,
and self-driving cars.
Yes, and that. Somebody's got to slow down. Somebody's got to speed up. Everyone's got to stay together. You're going to create traffic.
Yeah. You're going to get traffic without a jam. This is This Week in Science. Thank you for all those animal stories, Blair. It is time to turn now to Justin Science Corner. Justin, what you got?
International team of scientists has found that changes to the human oral biome during the shift from foraging to farming were not nearly as dramatic as those who have occurred in modern times due to antibiotics. They looked at 10,000 years of history, basically, from around the Neolithic up to the end of the Stone Age, or at the end of the Stone Age up to medieval times, and they etched out mineralized plaque, dental calculus. Did DNA checks on it, and they could tell basically what the biome of the mouth was. Interestingly, they were actually even able to track certain species of bacteria's emergence or sort of taking over or being eliminated from the mouth over time. But really the changes were pretty minor until we get to the modern age, and we have antibiotic use, maybe toothbrush starts getting used more frequently, toothpaste, that sort of thing. And now there's some antibiotic resistance in microbes, and there's much more significant evolutionary changes taking place within the bacteria of our mouths in the modern age. So this is going to be 22nd century problems. So I don't think we have to worry about runaway antibiotic resistant mouth microbes yet, but it may be coming. And then here's the one I'm going to end my segments this week with some terrible, awful news that has no silver lining and should give listeners a knot in their stomach as they realize that a living nightmare from which they cannot wake up is going to be the new normal.
And it has nothing to do with antibiotic resistance in your mouth.
Nope. That's going to be 20 seconds.
Okay.
If we get there, for those who get there.
Okay.
This is the global approach to climate change has so far been, say you're going to fight it, make some bold policy change suggestions, stick to a few of the easier to reach goals, congratulate yourself for trying. Meanwhile, according to IPCC researchers, Earth's vital signs have continued to deteriorate. Researchers, part of a group of more than 14,000 scientists who have also signed on to an initiative declaring a worldwide climate emergency, said that governments have consistently failed to address the root cause of climate change, which is the overexploitation of Earth. Since a similar assessment in 2019, they noted an unprecedented surge in climate-related disasters. Floods in South America and Southeast Asia. Record-shattering heat waves and wildfires in Australia, the United States, and devastating cyclones in Africa and South Asia. They apparently put this together before the unprecedented flooding in Europe, but that will be next year's report, no doubt, alongside something even worse that we didn't even realize was going to happen, which is also going to be new normal. Of 31 vital signs that they were tracking, key metrics of planetary health include things like greenhouse gas emissions, glacier thicknesses, sea ice extent, deforestation levels, that sort of thing. They found that 18 of those 31 hit record extremes for the first time in this past year. For example, despite the dip in pollution, thanks to the pandemic of 2020 and 2021, levels of atmospheric CO2 and methane are at all-time highs in 2021. Greenland and Antarctica both recently showed all-time low levels of ice mass. Glaciers are melting 31% faster than they did just 15 years ago. Ocean heat and global sea levels set new records since 2019, and the annual loss rate of the Brazilian Amazon reached a 12-year high in 2020. Things are so bad in parts of the Amazon that the Intergovernmental Panel on Climate Change found forest degradation linked to fire, drought, and logging are causing parts of the Brazilian Amazon to now act as sources of carbon rather than removers of the gas from the atmosphere. Livestock, such as cows and sheep, are now at record levels, with a mass exceeding that of all humans and all wild land animals combined. We are now a planet of domestic food animals. If you came from another planet and looked at what's the predominant earthling, it's domestic livestock. Recent record-breaking heat waves in the western United States and Canada, researchers say there's mounting evidence that we are nearing or have already crossed a number of climate tipping points from which the idea is there is no return. These include melting the Greenland and West Antarctic ice sheets, which may now be irreversible on a centuries-long timescale, regardless of how or if humankind slashes its emissions. There is increasing ocean deoxygenation and warming waters threatening warm water coral reefs, upon which 500 million people rely on for food, income, and storm protection, let alone the oxygen produced to the planet you can breathe. Given these alarming developments, this is, say, the authors of the study published in the journal Bioscience. Given these alarming developments, we need short, frequent, and easily accessible updates on the climate emergency. The authors echoed previous calls for a transformative change within 6 years, eliminating fossil fuels, slashing pollutants, restoring ecosystems, switching to plant-based diets, moving away from indefinite growth models, and stabilizing the human population. If that last sentence, stabilizing human population, does not give your spine chills, you might not have a spine. That's saying we're breaking the planet to where it can't sustain any more people. Stop having children. They've also called for climate change education to be included in school core curriculums globally in order to raise awareness. Again, awareness is one of those things that even those that in the United States, where 72%, according to that Yale poll, believe that global warming is happening, they also only hear about it maybe once a week from the media, even though this is an impending crisis that is causing all sorts of problems. So anyway,
I think that's really important what you just said, though, because You know, we've been saying over and over every week on the show, tell your friends and family to get vaccinated, tell your friends and family to get vaccinated. That has replaced the usual echo that we talk about, which is talk to your friends and family about climate change. Because there's an expectation, right, that you're hearing about it enough. We don't need to talk about that right now. But that's exactly the kind of statistic that proves that you need to talk to your friends and family about climate change, because if they're not hearing about it from the media, they need to hear about it from you. And believe it or not, there is a push-pull there. And if everybody starts talking about it and is looking for that information, the media will talk about it. Why is everyone reporting 10 different ways on the same COVID story every day? Because people want to hear about it. If we can make it where people
it's impacting their lives right now.
Exactly. Yes.
Yeah.
If we can make it feel urgent and real and solvable, which again, COVID is, by the way, even though it's whatever. But anyway.
And so is climate change.
Yes.
Climate change is solvable as well. And I think that is
people want to learn about it.
People want to learn about that. But we also need to inform people. People need to be informed that that change, it can come from them, but they also have to demand it of corporations, manufacturers, politicians. The entire system needs to change. So it's maybe it is a big ask to do it in 6 years, but I mean, there's a lot of political will that has to be addressed there. But I don't.
So again, the political will is it's of
but if we can get
slight
manufacturers,
majority of
investors.
Money's make believe anyway. You want to get something done. It is.
It's make believe, but it gets people to do things.
Yes, I know.
It's why everything is done at all right now. Like, politics is driven by money, greed, and money. So if we just get people, give them the money to do the good things. Why can't we do that?
Yeah, I mean, fake meat alternatives, all of a sudden is like this multi-billion-dollar business. And it's because someone realized, hey, I can actually make this taste not like rubber. And people want it.
You know how they did that?
You know
how they did that? Yeah. They did that with science.
Yes, with science.
But there's exactly, so now there's this big push. And that's exactly what we need, is we need people demanding things so that then the people who have the unjust, undue power will make the right choices.
Yeah. Yep. And like you said, I mean, it's a well-known problem for humanity that we have a hard time connecting with things that are not affecting us right here and right now. Like, I'm going to put it off. I don't have to think about that right now. I'm not going to worry about packing until the night before I leave.
Uh-huh. Night before, morning of.
Morning of. This is This Week in Science. Reality sometimes is hard, right? But we have to face it, and I'm glad we're facing it together. Let's face the science. Let's face understanding what the world is really like and move forward. And you know, sometimes the world brings us very interesting science that can distract us a little bit for just a moment from some of those depressing real world thoughts. Like, do you ever think about what happens when you move your head from side to side really fast and you're looking for things? There's this idea in neuroscience that, like, if you're moving your head really fast or there's something in motion as you're moving your head, that basically your eyes are taking little snapshots and then piecing them together, that there are these little breaks in the actual pictures that the eye is taking. That's not true. It's not true.
Never heard of that. So good.
And it's actually, there is your eyes like smearing the light. And that is according to a new study out this week in Science Advances magazine. According to their abstract, or the abstract of the article about it, the in focus about, which is great. This is a vision story in a section of the magazine called Focus, which I find very punny. Visual information is continuously sampled from our environment, even as the eyes move, which helps the visual system create a stable view of the world. So when you're moving your eye, it actually, like this, it's smearing things. It's taking a constant picture, and those streaks across the visual field actually help the brain to create a continuous and stable view of the world that doesn't break up and break apart. And so we're able to be perceptive, be aware of the motion of objects in our field in addition to the movement of our eyes. But I find it very interesting that we used to think that there were these, well, our eyes do these saccade motions. When you move your eyes really fast, especially like if you're sitting in a chair and you get spun around in the chair, your eyes don't just track with your head. They go forward and back and forward and back and forward and back and forward and back. Yeah, bop, 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 bop. These saccades, yeah. And so because of those saccades, they didn't think that we got that consistent, continuous motion. But this new study by Schweitzer and Rolfs demonstrates that the motion streaks help the oculomotor system correct for variability in eye movements and help to build that stable view of the world. So even though the saccades are there, they're not really there in the way that our brain interprets it because the light never changes. Our eye is continuously taking that picture. So they used a novel high-speed projector and they manipulated a visual scene during eye movements. They had the eyes moving and but the visual scene also changed. And participants saw 6 small patches with unique patterns. One of the patches was designated as a target for the eye movement. You're supposed to try and focus on the target. And the pattern ensured the patch would generate a unique motion streak. And then they wanted to know whether the participants were unconsciously processing the motion streaks. So they rapidly shifted the position of the target while the participants' eyes were in motion. And as a result, the eyes landed on a location between the actual target and one of the other patches. Because the eyes were like, wait a minute, that moved. So the eyes didn't quite go where they were supposed to. And so once the movement was complete, all the patches were masked so that there was really no difference between any of them. So there were corrective eye movements that had to be made by the eyes. But all of these things together, the end story for this is what I told you at the beginning. Our eyes are taking a continuous picture of the world.
I love it.
And they're streaky and smeary.
I guess you would assume based on how the eye moves that it's like pasting a bunch of overlapping photos together.
Yes.
But it's really like taking a pano.
Yeah. Yes.
So something interesting, like if you watch ballerinas doing like their spins, they're picking a spot and then they're
focus on the
same spot as every time they come back to sort of stabilize.
That's so they don't barf.
Right. No, I get it too. It totally makes sense. But then like everyone's like the Winter Olympics will happen and then the ice skaters are doing that like blurry spin where they like sort of just disappear into this hub of a spinning top. How in the world do they do that? Because that right there, that seems like that would be just so utterly disorienting.
Well, it's training, number one, but it's also...
So they don't just know how... This is what...
the way that it's set up, though, because the eyes are not kind of going back and forth and taking these individual shots, if you would imagine, if they were taking the individual saccade-type shots, the ballerina would have a much harder time being able to find that target at the other, when she came back around 360 degrees, because the pictures being pasted together by the brain...
It wouldn't make sense.
It wouldn't make sense. It would be hard for the brain to make out, like, where should things be, and am I in the right place, and is this the right thing? But it's specifically because of the streakiness, that streak goes around, and so you still have a streak that continues from that target from where you started to where you're ending. So your brain is like, there's this line of continuance, and I've been following it. So that's how it helps with the stability.
Okay, that might be true. Now what I want to do is I want to blindfold some ballerinas.
Yes.
And set them spinning on a stage and see how much of it is vision and how much of it is just like physical body memory of if I do this, I do that, and then I should be looking in the right direction. Like how much is it visual cue? How much of it is just within...
Is it the body? It's all, well, oculomotor. It all goes together.
Just put lots
Eyes are
of
involved in
pillows
that.
on the floor so nobody gets hurt.
Yeah, lots of pillows. And my very last study for the night is about little baby dreamy mice. Little dreamy baby mice.
Oh, what is you, little baby mouse?
Yeah, so this is maybe different with the way that human babies work and our eyes work, because our eyes don't work for several months after we're born. Like, our eyes really they're not ready to be there. But a lot of newborn mammals open their eyes right after they're born and has to start making visual sense of the world because predators are around. A lot of precocious animals really have to do it very quickly. But these researchers publishing in Science out of Yale University this week suggest that there's a kind of activity in the retina of the eyes of little baby mammals. They did this study using mice that is like dreaming about the world that they will live in. Yes. So before they're born, these little mice have waves of activity from the neonatal retina. This is before the eyes have ever opened, before they've come out of the womb, before they're still in there, waiting to be born. And they have these waves of activation. And these waves of activation mimic actual light-stimulated activity that would occur if the animal were moving forward through its environment.
Wow.
But how could this be?
How could this be? This dreamlike eye activity?
Is this something that's being provided somehow? Is this something that just was happenstance and gave an evolutionary advantage? This seems insane.
Right. I mean,
it's practice.
It's practice. It's like the ballerinas, but it's before practice. It's practice. But this is the interesting aspect. And I want to know, this is in mice. I'd love to know if this is something that happens in neonatal humans as well. Mm-hmm. But, and I'd love to know if there's a different stage of development at which this is present in different species depending upon their precociousness. So which species of mammals have this activation, this preparing for use? And when does it occur? How early does it occur?
But again, how? Like, what is the mechanism? This is killing me. I'm so confused.
I don't know, but this is
tell me.
This is a huge question, right? The why is so that the animal's more prepared to view things in the world and to survive. But
Warming
the how,
getting it ready for how it's going to be.
How does it know that the eye is going to need to have these kinds of signals in it to move through the environment? This must be evolutionary.
It's
evolutionary evolved, but yeah.
Is it a genetic memory? Is because we know the mom can kind of provide
what triggers it?
Weird memory-based biology to the baby. I don't know.
So remember, it seems like we always seem to have this sort of boxed, disconnected idea that the unborn child is removed from all life. It's like an alien showing up on planet Earth for the first time.
Like
I don't even know how to breathe. What am I doing here? What is up? But they are part of a billion-year-old life form that has been surviving on this planet. Now we pod off, we bud off into these separate individuals that are so different from each other and think we're unique. But ultimately, we're all the same organism that's just sort of mutated off into different directions and had a billion years of maneuvering on the planet. So somewhere within all of that information of existing on the planet, the eyes know what it's going to be facing.
And the brain as well. And so this is part of it, which is the brain before animals are born already has started firing and wiring and starting to put things together. And so this is probably part of that process of the the amacrine cells within the retina firing and trying to wire with the brain. Mm-hmm. It's probably setting up those neural circuits for success. And what makes me say that is that the Yale team also removed or stopped. They blocked the function of these amacrine cells in the retina. And so the flow of activation did not happen in those baby mice, and it impaired the development of the ability of these mice to respond to visual motion after they were born.
So you said endocrine, yes?
Amacrine.
Oh, amacrine. But that's still a hormone-related system.
Interneurons.
Okay.
In the retina, so they connect.
Okay. My question is,
cells.
Are the embryos dreaming when the mom is dreaming?
Oh, I don't know.
Because this is now my follow-up, right? Is there some sort of signal getting pushed to that being to promote that?
Yeah.
Or is it separate? Yeah. I'm curious.
But to some great degree, too, that is the same being.
Right. Which is why it might be related.
Yeah. Well, they are related.
They are, but is it? Anyway, I don't know. It's the mystery of life. The miracle of life.
Mystery
of life.
Still a mystery. Mysteries.
Mystery of mysteries. Yeah, but it's really
this is Mario Alonso you're belting out there.
But the take-home message from this is that the brain, the organism, the baby is dreaming about being born, is preparing to be born.
I don't want to go out there.
No, it's getting ready. It's like, yes, let's go. I'm going to know how to see. I'm going to do all this stuff. I'm practicing. I'm going to see the bright lights. Bright lights, big city.
No, I was way more comfortable in the womb. Yeah, it was way better.
Yeah. Try again tomorrow.
Have we gotten to the end of another episode?
I think we have.
Woohoo! We did it. We've gotten through the science. We didn't talk about the very old sponges. Maybe you out there will
850 million billion-year-old sponges.
Billion-year-old sponges.
Yeah, like the oldest sponge life ever discovered, and it's in Canada.
Oh, earliest animal yet found.
At the top of a mountain.
890 million years old.
You know, rocks. Yes. Not billion, million, yes. Old life.
Oldest animal on Earth by more than 300 million years.
And really, they're just looking at these little structures, these little kind of what look like could have been filaments in rocks. There's like
little spicules.
Yes. It's amazing.
I didn't see the pictures of the sponge one. I pictured it like a sponge imprint on a stone.
Yeah, it's like a sponge imprint, but it's...
Yeah.
Yeah.
But it's the grippy...
Was it the yellow side or the scratchy green side? That's really the question.
Neither, Blair. Oh. All right, now we're really done.
Okay.
We've really done it now. Thank you for being here for the show. We hope you enjoyed it. Thank you to Fada for your help with show notes and social media, for helping with those YouTube trolls tonight. Gord, Arnlore, Hot Rod, so many others. I know I'm going to miss someone. Our moderators over on Twitch and on YouTube, other places. Thank you for modding. Identity4, thank you for recording the show. Rachel, thank you for your assistance and the editing. And I do want to say thank you to our Patreon sponsors. Thank you to Pierre Valazar, but Ralph E. Figueroa, John Ratnaswamy, Kira, Carl Kornfeld, Jen Myronik, Melanie Stegman, DeKramsta, Karen Tazi, Woody MS, Andre Bassett, Chris Wozniak, Dave Bunn, Vigard Shefstad, Hal Snyder, Donathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Maddie Parangor of Sharma, Shoe Brew, Don Mundus, Stephen Albrand, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Ronovich, Ked Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, John McKeed, Greg Riley, Mark Hessenflog, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Kevin Railsback, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood. This profile name is hilarious in the context of some other podcast. Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Mallory Sutter, Philip Shane, Kurt Larson, Mountain Sloth, Jim Drapeau, Sarah Chavez, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, Patrick Pecoraro, Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you for all of your support on Patreon. And if you would like to support us on Patreon, please head over to twist.org and click the Patreon link on next week's show.
We will be back broadcasting from our YouTube and Facebook channels and from twist.org/live.
Hey, do you want to listen to us as a podcast?
Yes.
Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, you can get your friends to subscribe as well.
Yes, and for more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twist.org. And you can sign up for a newsletter.
You surely can. You can
Or
also
ballet
contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, and Blair at blairbaz@twist.org. That's lots of ats. Just put twist, TWIS, in the subject line, or your email will be spam filtered into the spongio-seal of an 890-year-old sponge.
If there wasn't enough ats for you in the previous segment there, you can also reach out to us where we are at Twist Science, at Dr. Kiki, at Jackson Fly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned any
that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science, This Week in Science, This Week in Science, 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 This Week in Science, This Week in Science, This Week in Science, 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 This Week in 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 Science, This Week